What if a flower could make color without ever making any color? There is a plant whose petals shine blue at the base, and nothing in that petal is blue. No blue pigment. None at all. By the end of this, you will know exactly where that blue comes from. You will see why nobody could explain how a plant builds it. You will meet a wrinkle so small that light gets caught in it. And you will find out why one flower wears the shine while its close relatives stay dull. Start with the thing you already believe. You look at a red rose, and you assume there is red stuff inside the petal. And most of the time, you are right. Petals are packed with pigment molecules that swallow some wavelengths of light and bounce the rest back at your eye. Grind up that rose, and the red comes with it. The color is a chemical. You could put it in a jar. Now try the same trick on a peacock feather. Grind it up and the blue vanishes. What you get is a dull brown powder. The blue was never in the feather. It was in the shape of the feather. And that is the first idea worth naming. Structural color. Color produced by the physical architecture of a surface rather than by any pigment inside it. A soap bubble is the cleanest example you will ever hold. A soap bubble has no dye in it. It is water and detergent, transparent and colorless. Yet it swirls with pink and gold because its film is unbelievably thin. Light bounces off the top of that film and off the bottom, and the two reflections arrive slightly out of step. Some colors cancel, some colors reinforce. Change the thickness by a hair, and the color changes with it. That is why the bubble shimmers as it drifts. The color is not in the soap, it is in the geometry. Once you know that, you start seeing it everywhere. The metallic green of a beetle shell. The electric blue of a morpho butterfly. The oil slick sheen on a puddle after rain. None of those are dyes. They are stacks and ridges and layers built out of ordinary transparent material, arranged so precisely that they sort light by wavelength. Which raises a question people rarely ask. How does a living thing build something that precise? A butterfly's scale has ridges spaced more finely than the wavelength of light itself. Nobody hands the caterpillar a ruler. We can describe these structures beautifully. We can measure them, model them, and simulate exactly how they bend light. What we could not explain is how they get made in the first place. And that is where a small weed of a flower turned out to be the perfect witness. Hibiscus trionum. A modest cream-colored flower with a deep purple bullseye at its center. The kind of plant you would walk straight past in a field. Look at that purple base at the right angle and something odd happens. A blue sheen slides across it. Tilt the flower and it moves. The purple is a real pigment. The blue is not. The blue is structural color, sitting on top of the purple like a film of light. And it is not there for you. So who is it for? Bumblebees see further into the ultraviolet than you do, and this halo scatters light toward the blue and ultraviolet end of the spectrum. Earlier work showed that bees can detect it and use it. The shine makes them better at working the flower, faster foraging, at least in the lab. So the plant is signaling. The question is how it manufactures the signal. Put that purple region under a powerful microscope, and you find the answer written across the surface. Rows of tiny parallel ridges running side by side over the cells. That is the second idea to name, a diffraction grating. A set of parallel ridges spaced so finely that they fan white light out into separate colors. You already own one. Tilt the underside of a CD toward a lamp and watch the rainbow crawl across it. The disc has no pigment in it. It has grooves. The grooves do all the work. The ridges on this petal do the same thing at a smaller scale, on something alive and still growing. And here is the strange part. They are not perfectly ordered, they wobble. A flawless grating would throw out a hard rainbow that swings wildly with angle. The messiness in this one smears the effect into a soft blue haze instead, which happens to be exactly the part of the spectrum a bee is best at seeing. The imperfection is the point. So what are those ridges actually made of? Not the cell wall, the layers sitting on top of it. The cuticle, a thin waxy skin that covers every leaf and stem and petal plant puts into the air. You have touched it 1,000 times. That faint greasy shine on an apple is cuticle. It is a waterproof coat that stops the plant drying out. On most of this petal, that coat lies perfectly smooth. On the purple base, it wrinkles into rose. Same plant, same petal, two completely different surfaces, a millimeter apart. And the mystery that had been sitting there for years is smaller and sharper than you might expect. Why does the wrinkling stop exactly where the purple stops? It gets worse. Some hibiscus species have the ridges. Others grown side by side in the same greenhouse have a base that is completely smooth. Same genus, same kind of petal, same purple pigment. One shines and one does not. If you enjoy watching a mystery get peeled apart one layer at a time, subscribe and stay with me. This one still has its best layer left. So what would you check first? If a surface wrinkles, the obvious suspect is a squeeze. Buckling. What happens when a thin layer gets pushed from the sides and has nowhere left to go but up and down? Push a rug across a floor and it rises into ridges. Squeeze the skin on the back of your hand and it folds into lines. Nobody drew those lines. The pressure did. The leading idea was that petal ridges form the same way. The cell underneath stretches lengthwise as the flower grows. The cuticle on top keeps getting produced. If the coat is made faster than the cell stretches, the coat runs out of room. Too much material for the space available. So it buckles. That gives you a clean and testable prediction. Two numbers should decide everything. How fast the cell stretches and how fast the cuticle is laid down. Get that ratio right and you get ridges. Get it wrong and you get a smooth surface or a mess of random wrinkles. It is a lovely model, simple, physical, and needing no genetics at all. You can see why people believed it. It also does not survive contact with the actual plant. Testing that means more than looking. It means reaching into a flower and changing it while it is still being built. And that turned out to be the real obstacle. The plant's biologists know best. The ones with decades of genetic tools behind them make no structural color at all. The famously shiny ones are tulips and orchids and tropical trees, slow and awkward, some of them taking years to flower. So the researchers behind this had to build a new laboratory plant from scratch. Hibiscus trionum was chosen for boring reasons, which are usually the best reasons. It goes from seed to open flower in about 7 weeks. It pollinates itself. It grows in almost any conditions you hand it. 7 weeks against the years a flowering tree needs. That gap is what makes an experiment possible at all. They also worked out how to insert genes into a species nobody had ever transformed before. Only then could the real questions begin. 2 possibilities sat on the table. Either the ridges are a side effect of how cells grow, pure mechanics, and the plant barely controls them. Or the plant is choosing, deciding where the pattern appears, down to a boundary a few cells wide. One of those answers makes a flower a passive lump of physics. The other makes it something much closer to a factory. So what happens when you force a petal to grow wrong? They dosed young buds with plant hormones and with drugs that wreck the internal skeleton of the cell. The cells came out wider, longer, different shapes entirely, and the ridges formed anyway, every single time. So they widened the search to the whole family. 8 different hibiscus species and varieties, 5 with striated bases and 3 completely smooth. They measured the cells and the cuticle in all of them and fed the numbers into the model. The model said every last one should have ridges. 3 of them do not. The model was not simply imprecise, it was missing something. The missing piece showed up in a gene. 3 related genes control cuticle production in this plant, and only one of them behaves strangely. 2 are barely switched on. The third fires up only in the purple region, right as the ridges begin, reaching roughly 200 times the level of the other 2. 200 times more in one patch of one petal, at one exact moment. That is not a side effect. That is an instruction. So they broke it. They engineered plants where that gene was altered, and the blue sheen at the flower center simply disappeared. Under the microscope, the base was smooth, bare, no ridges anywhere. And here is the result that killed the old model. Those cells were the same shape as normal cells. The cuticle was the same thickness. Everything the model cared about was unchanged. Only the chemistry had shifted, so they went hunting for it, sampling the molecules sitting on the petal surface without disturbing the layers underneath. The split was clean. Every striated surface shared one chemical signature. Every smooth surface shared a different one. Shiny petals were rich in waxes, and in one particular building block of the cuticle, smooth petals were loaded with another family of compounds instead. That pattern held in the engineered plants, and it held across the wild species too. Chemistry predicted the shine. Cell shape did not. Then came the test that settles it. Take a young petal before any ridges have formed and squeeze it sideways. In a normal plant, ridges spring up under your hands. In the altered plants, that same force does nothing. The surface stays flat. Same squeeze, same shape of cell, same thickness of coat. One surface buckles and the other refuses. And that answers the question we opened with. The wrinkling stops where the purple stops, because the plant stops making that particular chemistry there. The petal is not one material. It is two, painted onto the same surface in the right places, and only one of them is capable of folding. One caution before you run with that. This was shown in a single laboratory species using direct genetic manipulation, so it is not proof that every flower on Earth works this way. Although the same chemical signature turning up across 8 separate hibiscus varieties is a strong hint that the trick is far older than this one plant. Step back, and the interesting part is not the flower. It is the manufacturing method. Almost every color in your house is a pigment, and pigments fade. Sunlight pulls them apart, molecule by molecule. A color made of shape does not fade. Grind it and it dies. Leave it in the sun for a century and it holds. There are already paints and films and fabrics being designed to work exactly this way, colored by texture instead of by dye. The hard part was never the physics. It was getting a material to fold itself into that pattern without a machine pressing it. A weed in a field has been solving that for millions of years. It never presses the surface into shape. It changes what the surface is made of and lets the folding happen on its own.